Hi, I'm Devon Payne, and I'm going to be talking about the reductive dissolution of pyrite by methanogens and its physiological consequences. So we've shown that methanogens can reduce pyrite by growing these cells with either pyrite or canonical forms of iron and sulfur, and we see that when the cells grow with pyrite, they reduce it and produce sulfide that's measurable in solution in our biotic cultures, but not in our abiotic microcosms. And this research was published in the ESME journal this year, where we also showed that methanogens require direct surface access to pyrite to grow, where when cells are provided with pyrite that's free in solution, they grow very well. However, when pyrite sequestered in dialysis tubing, we see no growth, and this finding was further corroborated by our observations of cells growing in close contact with the minerals. This growth on pyrite has physiological effects at the level of cell size and the amount of iron per cell, where pyrite-grown cells are smaller and accumulate more iron than ferrous iron and sulfide-grown cells. This research was recently accepted in the Journal of Bacteriology. With that, I would like to acknowledge my mentor, Dr. Eric S. Boyd, our Boyd Lab members, collaborators, and our funding sources, which includes the Department of Energy. Thank you.